Yeah, so as Ollie said, this is all about actually making potentially useful things up in space. And I think it will be good to start with a description of the vision here, which is I guess a question for all of you. And in this in answering this, also tell us a bit about you and your own work. What can you make in space? Or what can you make in microgravity that you can't on Earth? Give people a bit of a hint of what we're going to talk about. We'll go into the details later, but maybe give us a sense of it. Maybe start with you over there, Robert.
So, hi everybody, Rob Garmeis, Director of Formulations and Exploratory Biopharmaceutics at Bristol Myers Squibb. We are leveraging microgravity to better understand our crystal structures of our drugs that we're making for both large and small molecules, and taking that information and applying it to our formulations to help patients on Earth.
Great. Hello, everyone. I'm Katie, co-founder and CEO of BioOrbit. At BioOrbit, our aim is to build a pharmaceuticals factory in microgravity to change the way in which antibody drugs can be given to a patient. So this is all about mass-producing crystals of antibodies that fundamentally allow for highly concentrated doses that patients can then self-inject rather than needing it in the vein. So we're looking, again, based on the crystallization and the benefits of crystallization in space, but for the mass production of drugs.
And Thomas, how about you?
Yeah, Tom Marshburn, Chief Astronaut and Vice President of Human Factors Engineering for Sierra Space. We're building an infrastructure in low Earth orbit to do all of these things and a way to get them back to Earth with their Dream Chaser spacecraft, launching for the first time for cargo to the ISS in May of next year. And I would almost turn around and say, what can you not do in microgravity? What's not changed? If we remove something as fundamental as the effects of gravity, both on a molecular and a macro scale, a lot of things change. How cells respond, how molecules come together, how colloids come together. We've only begun to touch what we can do in microgravity.
Let's go into that a little bit more then. Maybe Katie, maybe you can tell me, what is it about microgravity that allows you to grow crystals, drug crystals in a particular way? Just make it come alive for us, because obviously we've been doing drug manufacturing on Earth for a long time. It's been very good. What is it about space that allows you to do it differently and better?
Yep, of course, great question. So this all comes down to the fundamental physics. So when a crystal's growing, small amounts of heat are given off, which on Earth cause little convection currents. And then when the crystal gets to a certain size, sedimentation causes it to move and drop to the bottom of whatever it is you're growing it in. And fundamentally what that means is that you've got a lot of motion around that growing crystal. That means the next molecule finds it harder to get in the right position. So you get imperfections and a wide range of sizes. But if you do the same thing in space, no convection currents, no sedimentation, so the next molecule can get in the right position, so you get much more perfect crystals compared to on Earth. And
more perfect crystals mean better doses, or how does that translate into drugs that people use?
Yeah, so it means that they're much more reproducible. So the problems with imperfections and a wide range of sizes is that they're not reproducible. How do you track release rates, etc.? But by having uniform, like perfect or near perfect crystals, you then can track release rates. You do have reproducibility, and that's what's needed for pharma use.
Okay. And Tom, just again, for the people whom this is a new topic and maybe wants to get to understand the science of it, how do we know that this is true? How do we know this works?
Well, we've seen it. I gave an example in the last talk, Keytruda. It's a drug, samples that were grown of that crystal. It's a cancer drug that were grown on the ISS, came back, and you see these large, perfect crystals, as opposed to what looks sort of like smudges and dust because of the effects you were talking about. So we've seen it actually on a more macro scale when we're starting to talk about, we haven't talked about stem cells or ability to grow macro cellular structures like organoids as well in 3D. Similar processes, but to include sedimentation and pressing of gravity. So something it wants to, a living tissue wants to lay out, say, in a petri dish, but in microgravity can grow in an organ like it naturally does, say, in a fetus, and develops tissue differentiation. All that happens, and we've grown organs the size of my thumb up there. So things that you can actually see that have happened.
So a lot of this work, which you're turning into potential companies and products, it's come from basic research that's gone on in the International Space Station and other places, I suppose. Which, if I think about it, it's been going on for some time. People have thought about these things, but to turn it into a company or make it commercial is impossible because the launch cost was so high. Right now, we're in a different world. So maybe, Robert, you can talk about this. You work for what we might call a traditional pharmaceutical company. When did space become an interesting thing for your company?
Yeah, so I think BMS has a relationship with the International Space Station National Lab, and then before that, even the shuttle missions, where in the mid-90s we launched experiments on a shuttle mission to look at the production of antibiotics from bacterial cells. And they were able to show that there was an increase in efficiency of producing those antibiotics in the microgravity environment when you remove those sedimentation and convective forces. Most recently, the relationship that my team has had has been mostly on crystallization, first of large molecules. So leveraging, just to add on to what Katie and Tom said, we were looking at using microgravity to make more uniform size and shape particles for suspension. We had another molecule that tended to form needles, and that's not ideal when processing, so trying to improve the morphology of those particles to make them more processable for filtration or for drug delivery. And then also it could potentially allow you to form crystals for things that you're not able to crystallize on Earth. And so over the first two missions, we did take a couple of shots on goal with that, compounds that we weren't able to crystallize with the hope of trying to get a crystal structure in space. Unfortunately, they weren't successful, but that just sort of confirmed that those particular molecules were extremely floppy and very mobile, so it's very difficult to get them to line up. So moving forward, we just had a recent mission that launched in the beginning of November that's hopefully coming down tomorrow night, our first foray into small molecules. So looking at, again, using microgravity to make particles that are easier to handle for our production, and also to get some more information to learn about the molecules, to understand the risks that they have with physical and chemical stability in order to make better, more robust drug products for our patients.
So you're sending up your own missions basically now. Has that been going on for some time? I mean, I'm just curious is it because of the costs going down? Essentially, is that what it is?
So the first two missions were through grants that we had applied for through the U.S. International Space Station National Lab. Looking at the first mission was focused on monoclonal antibodies. 3 different monoclonal antibodies that we had sent up. And then the follow-on experiment for that was diversifying the molecules, so looking at different modalities of our large molecules from our portfolio, and then also designing iterative experiments where we screen conditions where we generate crystals, and then do a follow-on experiment taking that data live and scaling them up to 1 ml size. So we do our screening at a 2 microliter droplet size and going up to 1 ml size. The most recent mission was actually we were working with Redwire, who had their own grant through ISSNL that we collaborated with them. We provided the molecules for them using their hardware.
Okay. Tom, tell me about some of the other things you hinted at. We've been talking about crystallizing drugs, but what were the other sort of biological, drug-related, human biology-related things you can do in microgravity that are becoming feasible?
One thing that we haven't talked about too much yet, but is related to drug therapies, is what happens to cells, living cells. Their cytoskeleton actually, when offloaded from in weightlessness, actually there is different RNA and DNA expression. Typically, you get accelerated disease processes. So you send up a cancer cell, you get virulent, might not be the right word, but a very aggressive one. So you're able to much faster test out drugs against not only cancer in general, but someone's specific cancer, if you brought that up with you. And with enough volume, we talked about realizing revenue if you reduce the cost, not just of launching, but the amount of space you have when you're up there. If you're not constrained to a rack or competing other things that are going on, say that's as true of the ISS, and make it so the return is not only less expensive, but also very soft and gentle, so your return is greater. All these things contribute to increased revenue.
I guess also just in terms of the logistics of how you might do this, you can't send up dozens and dozens of researchers to be doing pipetting and all sorts of other things
Yep.
up in space. So does this all have to be automated? Is that how it works, Katie?
Yes, so we're going an automated route, exactly that. We don't want to have to depend on humans. For the scales, we've got two infrastructure loops that we want to use. One is through the re-entry vehicles. So you send the whole manufacturing kit up, crystallization takes place, you bring the whole thing back down. But the numbers get really interesting when you have permanent facilities on commercial space stations. So where you're renting space, you have your manufacturing payloads there. All you have to then take up are the consumables, but to replenish the system. So that's what we're designing for, so that we have at that stage, minimal human interaction just to replace the consumables, but that's it. We don't depend on them for production.
And Tom, is that the same for you?
Oh, yeah, yeah. Certainly humans have helped define what can be done with the pipetting, and humans can repair, et cetera. But our entire intent is to have robotically enabled payloads on our station or inside our Dream Chaser as well. And then again, the return, what I would suggest is that the scale of what you're thinking could be much greater, because the real estate that's going to be available in microgravity is going to be so much more than it is currently.
Yeah, so just to share some of our experiences. So on our first mission, when we sent up the monoclonal antibodies, we had a very manual process. So there was a pipetting into the protein, then you add the reservoir solution to initiate the crystallization. And we were fortunate enough to have Kate Rubens as our astronaut executing. She's a microbiologist. We allocated 3 hours for pipetting and setting up, and she knocked it out in like 45 minutes. And she was like, "Do you have more things for us to pipette?" And the second mission, Frank Rubio was executing our experiment, and he doesn't have a scientific background. So for him, he was struggling a little bit through it. He had gone through some pipette training, but very limited compared to Kate. And when we look to conduct experiments, especially a high volume of experiments, removing that interperson variability based on their skill level is really important to ensure quality results.
And I cringe at how long you had to wait and how expensive it was to get through the process as well. So reducing that cost and reducing the time to realizing an experiment on board is something we hope to achieve as well.
Just continue with you, Robert. You talked about how you've done various experiments over the decades, sending things into space, working out how things are growing, how to use them. But behind you, there's a big company selling pharmaceuticals on a regular basis, researching them in labs across the world. How do you integrate this completely new way of doing things into your sort of typical pipeline for a new drug? I mean, at the moment, I'm assuming no drugs grown in space or made in space are being put into humans. There's a whole regulatory process that goes on. I just wonder, what's the process from here? If you can prove that it's cheap enough, which is the first step, or I'd like to know how close you are to that. How do you integrate something like this into your manufacturing process?
Yeah, so I would say at this time, we're really using it as a research tool for our drug discovery to identify good drug candidates and taking that information and feeding it back into our pipeline. For us, I think one of the challenges that we had has been the cycle time. So our first mission launched in December of 2020. By the time we got the grant was written in middle of 2018. So 2 years to launch. The data came back. July of 2021, we submitted a follow-on grant that launched in March of 2023. So the cycle time is actually very long. So right now, it's not practical to say, oh, we have this compound in early development that's moving very quickly into development. Let's plan to send that up and test that. And I think that's where the commercial space stations, that'll be beneficial to us moving forward. At this time, we're still optimizing the processes to understand the conditions and the experiments in order to generate the data. Right now, we have to basically change the way that we do the experiments in order for them to be executed in microgravity by the astronauts. And so part of moving forward is understanding if there's automation out there or hardware out there that better replicates what we have in the lab. Because the processes that we have in the lab, we've run thousands of compounds through those processes. So we know how those data fit in comparison to other molecules. When we get some data back from discrete sets of experiments, it's hard to see how that integrates with historic data and being able to leverage that. So for us, we view this as a tool moving forward for us to identify the key risks for our compounds and help us design the formulations, or say that there are going to be challenges that we can't overcome, maybe it's best to look for another molecule to bring into development as a drug.
Katie, can I ask, which bits of the sort of drug development process, as it pertains to sort of space and microgravity, are you trying to make easier for drug companies? What is it that you can offer that stops someone like Robert having to think about every part of the process and from scratch?
Yeah, exactly that. So our aim is to sit next to Big Pharma to then make this all much easier. We have the hardware, we know we have our own pipeline so that we could work with you and say, all right, which drugs are you interested in? Leave it with us. We'll get all the chemical conditions sorted, do the iterations in space. These are the crystals that you can achieve. Now let's go into mass manufacture. So then you don't need to think about the hardware, etc. That's where we are. We've got that sorted. It's optimized for space, etc., etc.
And I just wonder, when you get to the point where you think, okay, this is a drug that works better, well, once you've created it up in space and the crystals are better or whatever else, and it's tested and trialed, you think, right, this is what we want, then the manufacturing part comes next. And that requires a whole new manufacturing setup, right? And I just wonder, Tom, what's the sort of vision for that? How far are we away from like a routine? What does it look like? I just can't even imagine it in my head. Is it just constant launches and things falling to the ground, like drugs literally falling from space constantly? Well, what does it look like?
That's a great question. I believe in the case of crystals, you can bring one back and then grow similar, if
So
not
you don't need to do the
exactly
whole
similar.
thing in space all the time.
So you don't have to do, in all cases, you don't have to do the whole thing. You bring back that seed crystal that then grows your what you need on the ground. That might be true of stem cell lines as well. But yeah, I mean, with the Dream Chaser, the ability to, it's agnostic to the launch vehicle. Launch, dock, and whether it's crew tended or not, loading it up and bringing it back. The Dream Chaser can, each one, we're expecting about a 30 cycle lifetime. And so to be able to come back and very quickly land, say, in the UK, land at the company's facility and just immediately offload with pristine crystals.
Yeah,
That's what it would look like, yeah.
I imagine it's cost-wise still very high. What's your sort of medium-term view of all of this? Maybe we can ask you all, because I think this is going to be the crucial question, isn't it, Robert?
Yeah, I mean, I don't know if I can comment on the cost as of yet, but certainly
It's definitely more than you'd normally spend on creating things
exactly.
right now.
And some of the and so I know Paul Reichert pretty well from Merck, who's working on the Keytruda project. One of the things that he's championed and I fully support is the potential for a pharma consortium, right? So as an individual company, would we be able to sponsor a low Earth orbit lab, right? Probably the cost for that would be very prohibitive. But if we have a consortium that we reach out across pharma and get everybody to chip in a little bit and then allocate the space and the time accordingly, maybe that's a more feasible model moving forward, right? So, and then, certainly with the cadence of increased launches, increased frequency going up and coming down, that makes it more sustainable and more attractive.
This all depends on reusable regular rockets, right?
Yep, and landing vehicles, yep.
Yeah.
Exactly.
So Tom, do you have a view on when this becomes economically interesting? I mean, it's definitely some point in the medium term, I guess.
I'll say that I'm not the best person to ask by any means, but medium term, whatever that means, that makes sense. Not tomorrow.
Less than a decade. Less than a
Yeah,
decade.
yeah. I mean, we want to realize it as soon as
Katie,
possible.
you've probably got a better idea of this then.
Yeah, sure. So I think I find it really interesting is that it's not as ridiculous as it first sounds when it comes to the unit economics, etc. when you're talking about such high value goods as antibodies. So antibody treatments are hundreds of thousands. It can be 100 at least, per dose. In Keytruda's case, I think it is around that, I think. So what that equates to is in the millions of dollars per kilo, can be tens of millions of dollars per kilo. So when you're creating that sort of order of magnitude and it's worth tens of billions per kilo, actually it's not as crazy as it first seems. I would also say that in our case, we're taking a drug that's already gone through FDA approval and we're forming the crystallized version of it. So the upfront cost to develop that drug has already happened. it's forming another product. Like, how can we maximize what's already gone into this drug to make one that can be administered in a different way? So it's like squeezing more juice out of the costs that have already gone in. Yes, that's a more expensive squeeze, but you can extend patents with that, so you can prevent billions in loss. So it's very interesting. In terms of what length of time, I mean, I think mass manufacture 24/7 production, that will be on the order of, 7 years-ish. But I do think that there is a lot more that's going to be happening in the 3 to 5 years time.
Can I ask, given the example you've just given is of an antibody that might be $100,000 or per treatment or more, is this going to be limited to very expensive drugs for now? I mean, you're not going to be making paracetamol up in space, are you?
Yeah, I think it's then looking at what benefits, like what are the benefits of it. And for now, it only makes sense for those really high value drugs. As launch costs come down, infrastructure costs come down, there will be more and more drugs that go over the threshold, because that threshold is going to keep decreasing.
Yeah.
So prove it out for the high value ones, and then you've got your pipeline sorted.
Yeah, that makes sense. And I guess that Tom, in your examples, have you got particular drugs, treatments, biologics that you're particularly excited about on that front then?
I'm excited about an entire range, keeping in mind that we're not just talking about pharmaceuticals. Company just signed agreements with Astral Materials and Space Forge, so semiconductors, pure, faster computing power. I think all of these examples, there's some examples of vaccines that have been developed. As it turns out, some bacteria are much more highly virulent, and therefore the development of the vaccine was much faster. I'm excited about the aging process, because space essentially accelerates the human body's aging process. Our bones, our muscles atrophy, the radiation has an effect. And what we can learn so much more about these, say osteoporosis. In 6 months, I would have developed bone loss equivalent to what would take decades for me to get if I had osteoporosis. So you can come up with treatments that are just very rapid. So I'm a medical doctor by background, so I think of more of the clinical aspects, that end product. But we're just starting to lift up under the cover of what we're going to find out.
There's a question from the audience. Here it is. It's what is the typical cost to BMS of running an experiment in space? I'm afraid I don't know what BMS is. I'm hoping one of you does.
That's Bristol Myers.
Yes, there you go.
Yeah.
Cost to, of course.
Yeah.
Cost to BMS.
Yeah.
Then this is the answer question for you. Then, how long do these experiments take, and do you bring the end product back to Earth? You kind of hinted at answering this already, but why don't we answer that directly?
Yeah. So through the grant process, through the U.S. International Space Station National Lab, they cover the costs for the space on the rocket ship to bring it up, and the crew time to execute the experiments, and the cost to bring it back. So for BMS, we don't ask for any additional funding to cover our headcount or our materials or anything like that. BMS covers those costs. So we're just basically looking for a seat on the rocket and a little bit of crew time. How long do these experiments take? So the experiments, we execute them within a 30-day period. The actual crystallization process varies based on the compound. Some compounds crystallize within a few hours, and some take a few days. But all of our experiments would go up on a SpaceX Dragon and come back down after the 30 to 35 days that they're on station. The biggest challenge is the cycle time to write the grant, get it approved, get all the paperwork in place, and then to be able to execute the launch. That's been the bigger challenge for us.
Please do ask more questions on the Slido, so we will get to them as we can. But Robert, just following up on this, again, we kind of talked a little bit about this at the beginning, but I'd like to just drill down it a bit more. The fact that you can make better versions of drugs up in space is tantalizing. How do we actually know that they work better in people? Or is it that you need less of it? Or what is it about those drugs that then translates into direct clinical benefit or economic benefit for patients?
Yeah, so for the experiments that we're executing on the International Space Station, we're really getting a better fundamental understanding of our molecules, right? So getting a better crystal with a better crystal structure, we can really understand the molecular interactions between our drug molecules. And you can leverage that one of two ways, right? So if you want to crystallize the drug, then you would choose excipients that don't interact at that portion of the molecule, which would increase the likelihood that you'll be able to crystallize the drug. If you don't want the drug to crystallize because you want to drive up your concentration solution and stabilize it in that way, you would do the opposite. You would say, okay, this is where the molecules interact with each other to form crystals. So let's select an excipient that will bind to that portion of the molecule and prevent it from binding to itself and forming crystals. So that's really the mechanistic understanding of what we're trying to get out of our crystallization experiments at this time.
Okay, thank you. Another question from the audience. How good/efficient/generous is the EU and UK in offering grants versus the United States? Who'd like to answer this one?
I'm
Katie,
happy to go for it. So yes, I'd say, as we were speaking, grants take a really long time to write and you're not guaranteed to get them. So when it comes to startups, you can't rely on that to move. You have to have speed, and that's where you need you need commercial backing and you need VC backing to get you to move. So grants are great, but again, it's not a reliable source that you can rely on. I think it's different when you're talking about defense and space. There's a lot more options and opportunity there, but when it comes to biotech in space, very limited. You're going to have to start deviating away.
So your answer to that question is that it's ungenerous on both sides. In that case.
I'd say that it's not where you should be. Well, this is just our experiences. You can't rely on it. It takes too long. If you want to move quickly, you need capital.
Tom, do you have any thoughts on that?
No, I mean, I've looked to my colleagues to answer that. I haven't had to write a lot of grants. So
Robert,
but I
you've written
totally
many
believe
grants
it. I believe
either
it.
side of the pond.
Yeah.
Yeah, so certainly we don't leverage the EU and UK funding. Our grant proposals have all been with the US through the US ISSNL. I should mention, I do want to mention that I have a good team of people behind me that actually write the grants that provide the background data. As the principal investigator, I focus on the strategy and the connectivity between BMS and our external partners. And I have, like I said, a lot of good people across BMS from early discovery to in the commercialization group that help support this. We are a truly matrixed team, and a lot of our team members do this as not part of their day job, but as part of their scientific curiosity and wanting to do something innovative to help patients.
Just as we wind up, I'm curious about another thing, which is obviously we've been talking about pharmaceuticals and sort of expensive, valuable molecules and biologics and things, which is one market. But Tom, you've hinted at this already. There are many, many other manufacturing markets for materials and other things. I'm curious to know if what you're learning in this space can be used elsewhere. Is the basic technology and the sort of the logistics and all of that going to be useful for other things as well?
Oh, certainly. You mean beyond
biologics, beyond drugs, etc.
And drugs. Well, I mentioned the semiconductors. Of course, you have not only do you have access to a wonderful vacuum, you're in it, but also the lack of sedimentation. Your convective forces, you're able to grow very, very pure semiconductor crystals. I actually was involved in an experiment. I think it was the first time to have it done. I started drawing fiber optics, and I haven't seen the results of it yet, but I understand that it's grown. Literally, they were able to actually pull longer, thicker fiber optics cables, which are much more pure. So, and these are just a couple of examples. To give you an idea of the level of extent that we could go, there was one experiment I was working on that looked at capillary flows. So you could build motorless rocket engines. Turns out that fluids in space act like picoliter level volumes of fluids on the ground. So they actually took that technology to build smaller labs on a chip to develop an entire laboratory, put it in a box, and send it to places that don't have laboratories, and to do blood analyses. So my imagination is nowhere close enough to state all the things that are going to be coming out of this.
Katie, in 20 seconds, where else can you take what you're doing? Is your imagination wide enough to see where we're going?
Yeah, I mean, for us, again, the grand aim is to have our own space station in manufacturing all of these drugs, and that's a very normal part of the supply chain, and that it's not space, it's just manufacturing. So that's our aim.
Okay. Thank you very much, all of you, for that fascinating discussion. And can I have a round for my panelists?
Okay.
Thank you all.
Thank you.
Thank you very much.
Thanks a
Tom.
lot.
Appreciate it.
Appreciate it.
Thank
That was
you.
great. Thank you.
Thank you
Katie.
very much.
Thank you, Katie.
Thank you.
Thank you very much. Okay.